Arizona State University, and a team of biologists, Eric Thévenot and Fernand Duarte from Toulouse. So I'm going to talk about a packed cell tissue. So the problem is suggested by the biologists who are trying to understand the mechanisms that allow cancer cells to leave the tissue where they first appear and spread to other organs. So this is the first stage of metastasis that they want to try to understand. And for that, they use a biological model, which is epithelial tissue. And this tissue is surrounding all the organs. So the cancer, to be able to spread, it has to pass through it. And that's why this type of tissue is used to study this phenomena. And this tissue is constituted of closely packed cells. So the contact forces should play an important role in its dynamics. In particular, it has been studied also in the context of development, how shape and emerges and remains stable, and in cancer, how the effects on individual cells may lead to the formation of to the deformation of the tissue. So I am going to, so in these studies, people complement lab experiments with mathematical modeling, because it is not always easy to measure the impact of contact forces in the dynamics. So the models are used to test hypotheses. So that's what we are going to do as well. And we use simple cell shape. So we'll model a cell by a hard sphere. Yeah, because we want to keep it as simple as possible in order to be able to more easily connect causes and consequences of the behavior that we observe. So I'm starting to present in general the model, the general framework, and then I'll apply it to the study of the neuroepithelium in 2D. So, so consider a system of n particles under a force F. Then the position and the velocity is governed by the second Newton's law, given by these equations, and the dynamics would be smooth if there wasn't this non-overlapping condition, so the collisions. And in order to get insight about these dynamics, we discretize in time. So we use a time stepping scheme, and at each time step we perform 3 steps. First, smooth motion. So we simply use Euler discretization to evolve the particles. Then we are doing mistakes, so we try to correct these mistakes. And we do this by solving a minimization problem. So we assume that the system wants to minimize a potential energy subjected to non-overlapping constraints. And then we get a local minimum with non-overlapping. And finally, we update the velocities. So this is the general idea. Now the tricky part is this second step, because since the particles are convex, the non-overlapping constraints are typically non-convex, and this gives rise to a non-convex minimization problem that is not very easy to solve. So in this, so even if we consider a convex potential, as I said, the constraints are non-convex. In this case, phi KL is given by the diameter squared minus distance between 2 centers, and this has to be negative, so that the distance is bigger than the diameter. Yeah, in order to solve this problem, so the problem has solution because the potential is convex, and in order to solve it, we use the Dampf-Darrow-Hurwitz algorithm that we have proposed here and explored numerically. I will not explain now, but I can explain later if you want. So at this point we are working on the stability of the steady states of the system, which correspond to the solutions to the minimization problem. This study was mainly numeric, but so for now it's working. So we use it to model the epithelium. So I'm going now to talk about the model. First at equilibrium, then equilibrium breaking, and finally I'll give some examples on how defects could lead to different shapes. So the biological model is a chick embryon, and so they work with a cross section of the neural tube. That this is what gives rise to the spinal cord, and this is the cross section. What you see here are not cells but the nuclei, and as you see, it's very dense. The cells are in fact very stretched and very thin, so the volume of the cell is almost the volume of the nucleus. You can imagine a sock with a watermelon inside, and that should be a good representation for it. So the cell is connected to the basement membrane, and at the apical side it's connected to neighboring cells. The basement membrane is surrounding the tissue, and the apical side is in the middle. So we are going to use hard spheres not to model the cell itself, but the nucleus. And so with center xi and radius r, hard radius and soft radius, okay. So radius of the hard sphere and radius of the soft sphere to give it some flexibility. Then we use a point to model the apical point and the basal point that are connected to the nucleus through springs. And the rest length of these springs is the radius. So if a cell is alone, then it should minimize this potential, which is given by the energy associated to the springs. So the distance between A and X is R, and etc. And in this part here is alignment force. So we want that these two vectors are the cosine is -1. So we want A to be opposite to B. Okay. Now, if we add another sphere, another cell, we add to the potential the interaction between apical points through another spring, and also a non-overlapping constraint. We also impose non-switching constraint between the basal points that I forgot to say, but they live in 1D. And they cannot also be too far away, so there's like lateral adhesion. So this is the tissue at equilibrium, is a solution of this minimization problem. Now, if we now to introduce dynamics, the dynamics is driven by cell division and noise. So what happens in what we observe in the movies is that a cell that wants to divide, it starts to approach the apical side before division. So this is the dynamics of the tissue. Most of divisions occur at the apical side. So what we will do is we'll change the rest length of the springs such that the nucleus wants now to be here, near the apical point. Also, right before division, it gets harder, and finally it divides. So each cell has an internal clock where it starts to go up so we change the properties of the springs at this point, and then it divides. So the algorithm will have this will be like this. So first we generate the lifetime and the birth hour for each cell. Then we initiate the system when all cells have been born, and at each time step we divide the cells that are ready to divide, increase the rigidity of mitotic cells, actualize the springs of cells that want to go up, and introduce noise, and finally use the minimization algorithm to make the system non-overlapping. So I will now quickly show a video. So this is real tissue. Here is the basal membrane, and on the left is the apical side. And you can see that the before division, for example this one, it goes there and then it divides. So, and it's quite quick, this movement. Although the tissue is very packed, they manage to find a way through the crowding. The noise seems to be also important in this. In our simulation, we have here the basal points, and here on the top the apical points. In blue, each cell has apical, a basal, and the nucleus. In yellow, the nucleus is trying to go up, and in red, it's division. So we see that it seems to be doing what we want. The nucleus seems to approach the apical part before division. So qualitatively, it seems okay. Now the aim was to study the defects. So what happens when the polarity or the proliferation or the basement membrane is degraded. So this is okay. I don't have much time now but in any case, these are only preliminary results, because the next stage now is to validate the model with a healthy tissue, and then we'll be able to study this situation. So just to give two quick examples, in the case of the polarity that we model with the alignment force, if we remove it, we observe the cells coming down to the basal membrane and the apical points as well. And this seems similar to what happens in the cancer case as well. Another phenomenon is if we increase the proliferation rate, so we decrease the lifetime of a cell. We observe that the divisions do not occur apically anymore, but they occur in the middle, and this is in agreement as well with the experiments. So yeah, it seems a good way to go. So just to summarize, so we are trying to understand the interactions behind metastasis, and the modeling framework uses a simple cell representation based on a minimization problem. This framework has been applied to the study of neuroepithelium and seems to be producing a good agreement with data, but at this point we still need to validate with a healthy tissue, then use it to study the cancer tissue, and this is ongoing work. Then eventually introduce more complexity, for example, coupling with a fluid model to take into account interactions with the medium. Yeah, and this is all. Yeah, and I want to acknowledge the work in the algorithm, in the minimization algorithm by Pierre and Sebastian, and the application to the epithelial tissue by Pierre, Sarah, Fernando, and Eric. That's all. Thank you.
Questions?
I got a little confused.
Yeah.
You have hard spheres.
Yes.
I understand this. Then you have some soft spheres.
Mm-hmm.
Outer layer.
so it's
Is
like
there some physics? How do you describe the physics of the soft layer interaction?
So the idea is to make it more flexible because the nucleus is rigid, but it can deform a little bit.
Okay, but you're modeling this soft part somehow.
Yeah. So it's like I mean, it's if you I could model it as an ellipse, for example, that could deform, and that would be more realistic. But that's more difficult. So.
But what are the interactions?
What
Hard spheres interact in a certain way.
Yeah.
So don't you need to interact forces to model the forces for the soft?
Yeah, I didn't. Yeah, I mean the soft sphere interaction comes in the potential. I didn't write it actually, but there should be a fourth term here, and that's where the cells try to go away from each other, but sometimes, I mean, it's not hard potential, so they may overlap in the soft part. Sorry, I didn't add it there.
Quick one.
Yeah.
I didn't quite grasp whether this is all two-dimensional, right?
Yes.
Is this a good model? Is there any reason to think that there's some three-dimensional effects on the actual cells?
Yeah, actually, it's very interesting to, if we could then go to 3D, because at the apical side, the cells are connected to each other and they form like a network. And this is a 3D effect. I mean, it's difficult to model this in 2D, right? So that would be interesting to check that case, what happens. Also, there's more space in 3D than in 2D. So it's easier to go up, in principle.